Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this gluconeogenesis lecture, I will be explaining you energetics of gluconeogenesis and applied clinical aspects of gluconeogenesis. So I will be going over the ATP consumption in the conversion of gluconeogenic precursors into glucose molecule, and also I will be going over a pyruvate carboxylase enzyme deficiency. Now let's get into details. Now the energy required for gluconeogenesis, that is hepatic gluconeogenesis. So the glucose for other cells and tissues, where the liver which is making glucose only for the other cells, how many ATPs that liver is going to spend in order to make glucose. Note that gluconeogenesis will be going on under fasting condition and starvation. At that time, so there will be fatty acid oxidation going on the other side in the hepatocyte mitochondria, giving NADH plus and FADH2 and ATP generation going on in the electron transport chain. So those ATPs are going to support gluconeogenesis. Now, how many ATPs are needed to convert a gluconeogenic precursors all the way into glucose? So, as per the calculation, so there will be 6 ATPs needed for the conversion of 2 alanine into 1 glucose or 2 lactate into 1 glucose. So, the ATP needs of gluconeogenesis in the liver, it is met by fatty acid oxidation in the mitochondrial matrix. That's why always hepatic gluconeogenesis, it is dependent on fatty acid oxidation. Now the table here is depicting why 6 ATPs are needed to convert 2 alanine into 1 glucose or 2 lactate into 1 glucose. So the reactions are written here. Pyruvate carboxylase enzyme, which is converting pyruvate into oxaloacetate. That means we need 2 pyruvates to make 1 glucose. So 2 pyruvate into 2 oxaloacetate. So by pyruvate carboxylase reaction, we need 2 ATP there. So that step needs 2 ATP. And then phosphoenol pyruvate carboxykinase, which is converting oxaloacetate into phosphoenol pyruvate. So 2 oxaloacetate converting into 2 phosphoenol pyruvate, where there is a requirement of GTP there. So GTP equivalent to ATP in terms of energy. So 2 more ATPs there. So 2 GTPs written in the table also. Now, phosphoglycerate kinase step in the reversal reaction of glycolysis, where 3-phosphoglycerate is converted into 1,3-bisphosphoglycerate. That is, in that step, since 2 molecules are converted here, again you need 2 ATPs there. So like this, 6 ATP molecules are required for the conversion of 2 alanine into 1 glucose or 2 lactate into 1 glucose. Remember that, and also remember ATP needs of gluconeogenesis is supported by beta oxidation of fatty acid in the mitochondrial matrix. Now, gluconeogenesis provides glucose needs of brain and red blood cell. That's why the gluconeogenesis is going on under fasting condition, and gluconeogenesis during fasting state clears lactate, glycerol, propionate from the circulation, because accumulation of lactate and propionate especially may lead to acidosis. That's why lactate can be cleared, propionate can be cleared from circulation during gluconeogenesis, making glucose and releasing that into the blood. Defect in the conversion of propionate into 𝐒𝐮𝐬𝐜𝐫𝐢𝐻 can lead to propionic acidemia and methylmalonic acidemia. So I have explained you all on this in my previous lecture on gluconeogenesis. And also uncontrolled gluconeogenesis will lead to hyperglycemia, like what is seen during chronic stress and diabetes mellitus. Note that whenever a person goes into a chronic illness, that chronic illness will release hormones like cortisol, and the cortisol will promote gluconeogenesis and will lead to hyperglycemia. And this hyperglycemia, it decreases immune capacity, and also it is going to lead to a compromised immune mechanism in the body and decreases the healing process, and also it will lead to so many other consequences overall, not allowing a proper healing process to go on. Okay, and that's why uncontrolled gluconeogenesis is not a good thing and it has to be controlled. And one of the cause for diabetes mellitus, especially type 2 diabetes mellitus, can be uncontrolled gluconeogenesis process, giving rise to excess blood glucose or leading to hyperglycemia. Now let's move on to see one of the disorder that is associated with pyruvate carboxylase deficiency. Now, pyruvate carboxylase enzyme, it is involved in the conversion of pyruvate into oxaloacetate, and this pyruvate carboxylase enzyme can be deficient, leading to decrease in the gluconeogenesis process. Not only that, decrease in the gluconeogenesis happens here. So conversion of pyruvate into oxaloacetate, as it is shown in the figure here. So this oxaloacetate, it goes into different metabolic fate. Oxaloacetate undergoes transamination reaction to make aspartate, and aspartate is necessary for urea synthesis, urea cycle. Aspartate is used in the urea cycle to neutralize ammonia. And in fact, it is ammonium ion. So aspartate is needed for neutralization of ammonium ion in the urea cycle. And also aspartate itself is a neurotransmitter, remember that. And also oxaloacetate getting into the TCA cycle, condensing with acetyl-CoA to make citrate. And citrate is needed for TCA cycle, and also citrate further down in the TCA cycle makes alpha-ketoglutarate, and this alpha-ketoglutarate will make glutamate. So note that glutamate itself is a neurotransmitter, and glutamate is needed to make so many other non-essential amino acids. Glutamate also makes GABA, gamma-aminobutyric acid, upon decarboxylation of glutamate. So gamma-aminobutyric acid is a neurotransmitter. Oxaloacetate is converted into phosphoenol pyruvate in gluconeogenesis process to make glucose. Okay, so this is another fate of oxaloacetate there. Like this, oxaloacetate that come from pyruvate carboxylase reaction goes into different direction to participate in variety of metabolic pathways. Now in pyruvate carboxylase deficiency, what will happen is, so I am going to block this reaction now. So pyruvate carboxylase enzyme reaction is decreased or absent. So what will happen? So there are two things that happens here. One is oxaloacetate will decrease, other is pyruvate will increase. Because of this, what is the consequences? When oxaloacetate is decreased, that means aspartate formation will decrease. When the aspartate formation decreases, that means urea cycle will decrease. When the urea cycle decreases, ammonia levels will increase. Okay, so there will be increase in blood ammonia level, leading to hyperammonemia. And this hyperammonemia causes neurotoxicity, and neuronal changes in the neuronal metabolism can happen, and that can lead to ataxia, chorea, athetosis, and other signs and symptoms. Now, citrate cycle, that is formation of citrate decreases, and that means TCA cycle decreases because oxaloacetate is down. When the TCA cycle is down, so that means ATP levels will be decreased because TCA cycle is one of the source for ATPs. And also note that alpha-ketoglutarate levels will decrease accordingly because alpha-ketoglutarate is coming from oxidation of citrate in the TCA cycle, and that will lead to decrease in glutamate, which is a neurotransmitter, and that can also decrease gamma-aminobutyric acid. Overall, a neurotransmitter levels will go down, and that can also contribute to neurological signs that we see in pyruvate carboxylase deficiency. So I'll just write it as neurological signs. Now, we have seen hyperammonemia. We have seen neurological signs, causes for neurological signs. And now, since oxaloacetate is down, so phosphoenol pyruvate is decreased. And when the phosphoenol pyruvate decreases, that means glucose formation decreases under fasting condition, and that will give rise to fasting hypoglycemia. So that is one of the sign you see in pyruvate carboxylase deficiency. Now on the other side, when pyruvate is not going into oxaloacetate side because of pyruvate carboxylase deficiency, so pyruvate is building up, and this pyruvate is now diverted into lactate formation. So lactate levels will increase, leading to lactic acidosis. On the other side, pyruvate can also be taken towards alanine formation by transamination reaction, and alanine levels will increase in the blood, giving rise to hyperalaninemia. Alright, so like this, so many metabolic consequences can happen with pyruvate carboxylase deficiency. Just to name few again, so urea cycle is affected because of lack of aspartate formation and leading to hyperammonemia, and hyperammonemia can lead to neurological issues. TCA cycle will go down, thereby decreasing ATP availability coming from TCA cycle, and also the intermediates of TCA cycle like alpha ketoglutarate, which is eventually making glutamate, will decrease, and that will also decrease gamma aminobutyric acid. Overall neurotransmitters will decrease, giving rise to neurological issues. And phosphoenol pyruvate formation decreases coming from oxaloacetate, that decreases gluconeogenesis process, and that leads to fasting hypoglycemia. On the other side, pyruvate is diverted into lactate and making lactic acidosis there, and alanine is also made from pyruvate, giving rise to hyperalaninemia. Now here are the signs of pyruvate carboxylase deficiency, especially the laboratory signs mentioned here. So severe fasting hypoglycemia, lactic acidosis, hyperalaninemia, even ketoacidosis, because some of the pyruvate converted into acetyl-CoA, and that will go towards ketone body formation, hyperammonemia, and changes in the neuronal metabolism leading to several neurological issues like hypotonia, ataxia, tremors, choreoathetosis, and also mental retardation. So these are all the signs of pyruvate carboxylase enzyme deficiency. Coming to different forms of pyruvate carboxylase enzyme deficiency that are known. One is type A pyruvate carboxylase deficiency, which is referred as North American phenotype. Other is type B French phenotype, and type C is a benign phenotype. Out of all these, French phenotype is the most severe one, and it will lead to death in the first decade of life because of so many neurological issues. So French phenotype is the most severe one compared to North American phenotype or the benign phenotype. So benign phenotype, in that sense, it will be a mild one. North American phenotype is a moderate one, and French phenotype is the severe one. So the severity accordingly, so given here infantile form, neonatal form, and benign form. So neonatal form is the severe form of pyruvate carboxylase enzyme deficiency. Now the laboratory features of pyruvate carboxylase enzyme deficiency. So patient will show fasting hypoglycemia, lactic acidosis, ketoacidosis, hyperammonemia, low aspartic acid, hypercitrullinemia, and hyperlysinemia. So these are some of the signs that we see in pyruvate carboxylase enzyme deficiency. Now let's look at pharmacological and dietary therapy for pyruvate carboxylase deficiency. So stimulating pyruvate dehydrogenase enzyme complex by supplementing thiamine and lipoic acid coenzymes can also help in the diversion of pyruvate into acetyl-CoA, because conversion of pyruvate into oxaloacetate is down. That's why we are one of the way to take out that the pyruvate accumulation going into alanine or lactate is to divert that into acetyl-CoA formation. Let me write down here. So the pyruvate has different fates. So pyruvate can go into lactate whenever it accumulates. It can go into alanine, and that's what happens in pyruvate carboxylase deficiency, leading to increase in lactate, lactic acidosis, increase in alanine, hyperalaninemia. Pyruvate, at that time we can divert that into acetyl-CoA, diversion of pyruvate into acetyl-CoA instead of going into lactate and alanine. So divert it. So basically by promoting pyruvate dehydrogenase complex enzyme, thereby decrease some of the pyruvate, thereby lactic acid, hyperalaninemia and other things can be decreased. That's one way to do it. So other thing is biotin supplementation. You know, pyruvate carboxylase enzyme here, it needs biotin as a coenzyme. So supplementation of biotin might optimize the residual enzyme activity, thereby improving the enzyme activity and improving the signs and symptoms. Citrate supplementation can be done to continue TCA cycle so that alpha-ketoglutarate, glutamate, all these things can be taken care. Aspartic acid supplementation to continue urea cycle can be done, and also to prevent fasting hypoglycemia, high carbohydrate diet or high protein diet to prevent patient going into gluconeogenesis, especially the cornstarch is the one that has been tried. So these are some of the pharmacological and dietary therapy that we can use in trying to decrease the signs and symptoms of pyruvate carboxylase enzyme deficiency. So that's all about energetics related with gluconeogenesis, and one of the disorder that is associated with gluconeogenesis enzyme, that is pyruvate carboxylase enzyme deficiency. I hope this video has helped you in understanding what all the number of ATPs that are required to convert a gluconeogenic precursors into a glucose molecule, and also the details on pyruvate carboxylase enzyme deficiency. Thank you.